We'll begin momentarily. I'm waiting for the benefit of our online audience. So I think most of you are here, hopefully everybody. We are broadcasting this for our online listeners. So just be aware there are microphones in the ceiling, so that everything you say is public knowledge and broadcast through the whole world. But don't let that intimidate you from asking questions, right? So we'll give them, just so that they get everybody's logged on here, we'll give them another 6 minutes and then we'll get started. Can you hear me okay back there? Good, okay. We'll try to talk loudly since we don't have a mic. Hopefully our online listeners can hear. I don't know, where should you at? We should have somebody monitoring the online just to make sure things are going okay. Maybe he'll be back in a moment. It'll speed my talk up. Yeah, I can. There's enough Macs in here to plug it in. Okay, I think I'm going to get started just a little bit early. So welcome to our online viewers. I hope you're able to hear. Please get a message to us if for some reason you can't hear. And we'll also speak up. Feel free to speak up at any time. I understand you should be able to speak up on the speakers here. So I want to go over the schedule a little bit. We're here, and I'm gonna talk briefly, just give the first little introductory about SAXS, and then Kushal is going to dive into planning and performing your experiments. I'm beginning to realize more and more that we need to compose a document on sample preparation. It's there's actually a lot to know about it, and that's why Kushal has devoted a lecture to that. So it's kind of unfair of us to ask you to prepare samples when you don't really know how to prepare samples yet. So, but this is a learning experience, and we'll try to improve our online hints and helps for doing this in the future. Yeah, I apologize we're going to be a little disorganized this time around. We've had, it's been an exceptionally busy time for us, a good time for us. Also, we have Barb, who runs the website, is out on medical leave too, so we haven't been able to update the website as easily as we would like to. So, so Kushal, who will give you that lecture, then I'll tell you a little bit more about scattering, and then we'll have a break, and then I'll do more about scattering. And then we'll have some lunch. Lunch is provided. And now there's a group photo at 1 p.m. So please be around for the group photo if you want to be in the photo. We're going to stand out on the deck there on the roof, and it makes a really spectacular. Unfortunately, there's no sun today, but maybe later. Okay, we have a photographer. So, okay, let's see. There are a couple other things I want to mention. Oh yeah, let me just finish the rest of this. Okay, group photo. Thomas Grant will be talking to you about envelopes later in the day. And then I have a very short talk. Jesse might actually give that while I run over to the beamline and finish some preparations on publishing SAXS data. And then Steve Meisberger is going to talk about size exclusion chromatography, which is quickly becoming the most frequently used mode of operation for collecting SAXS data. And then also we'll tell you, this is not an advanced course. The idea here is that most of you, how many of you have been to a synchrotron before? Okay, fair number, less than half, good. So our aim here is to introduce people who've never been to a synchrotron and never done SAXS data collection. And so we're going to try to keep the lectures at a basic level. That's why I call this BioSAXS Essentials, because this is intended to be your essential need-to-know information to get started, the first experiments that you do. And we try to focus on the easiest things that are the most powerful. So SAXS is a relatively easy thing to do, and there are some really straightforward things that you can put right into your papers without getting into a whole lot of sophisticated things like atomistic modeling. So, but we will talk to you about some about more advanced techniques, and Kushal will tell you about that. And then we're extremely excited to have Thomas Grant look. Any of you who might have been here last year heard some hints about this incredible ability to actually get real electron density directly from SAXS. And that paper, Thomas has published that paper in Nature Methods. It just came out, and we are proud to say that the raw software, BioSAXS raw, that you can download for free and that you'll be using to process your data here, has an implementation of Thomas's dense electron density reconstruction. And so part of your exercises tomorrow will be to calculate some electron density. So we think that that is really a game changer, and we're very, very pleased to be able to provide that to you. And it's I think, a rare opportunity where an algorithm has literally just come out, and the scientific community has hardly had time to see what it can really do. And so you have a chance right here to learn from the author of the paper and to actually use it. And who knows, you might actually see something that nobody's seen before. So we're very excited about that. Okay. I don't know what end of days lectures are. We'll talk about the nuclear holocaust. Okay. Oh, yes. So very important. I forgot to put on my list. So you will find on your memory stick some, and we also emailed you some software installation instructions so that you can get the raw software and all the associated modeling stuff onto your laptop. And there is a software installation directory on your memory stick.
Actually, it's just in the top. So tomorrow for the tutorials, it's hands-on, you're working on your laptop. So you need to bring your laptop, bring your flash drive, and please have the software installed ahead of time if you can. If you've tried to install it and had trouble, or you tried to install it during the breaks today and had trouble, let me or Steve back there know, and we can help you get the rest of it installed. We'll be running the tutorials tomorrow. So, yeah, and you also need the AdSAS software installed, which is in the instructions, but legally, for licensing reasons, we couldn't put it on the flash drive. So download that and install that as well. As I said, if you have questions, talk to me or Steve during the breaks today.
Yeah, so please try to, those of you who don't have beam time tonight, please make an effort to get that stuff installed, because it's always frustrating when installations don't always go smoothly, and we have limited person power here to help you solve installation problems. So the more in advance you know about them, you have the authors of the software here, so you have the best possible people in the world to ask about some of the software that's on there. So so give it a try, install it, and then if you encounter any difficulties, you'll be ready tomorrow to hit the ground running, and we can help you solve those problems. So, okay. Is there anyone out there in the online audience who has any questions? Or speak up, let us know if you can hear. Okay. I hope they can. They may be just waiting for the good speakers. Okay. All right. So tonight we had some. So I have a tentative schedule, and I realize some of you are not on here. And the two things that we need to do when we have a break later today is we need to look at the running schedule and get people on who aren't on already. And if the time is not a good time for you, we can try. We're very so if you want to sit in on someone else's data collection time, that's fine. Let them, control the show. But you are welcome to show up to any of the Beamline slots and sort of hang around and assist or whatever. So. And for those who didn't bring samples, we do have some basic standards which you can use to try data collection. So tonight, let me just explain. We have two beamlines, F1 and G1, and when you show up to your slot, go to the MacCHESS operations desk, right, which is in, I hope you can find, which is in the Synchrotron, and the operator will get you checked in if you aren't already checked in by virtue of registering here, and make sure you have your badge, your dosimeter, and show you where to go. The G1 station is the station that we normally provide when you come here in the future to sign up for time. G1 is the station that you will be running at. F1 is a crystallography station that we have temporarily hacked into a SAXS station. Now, it's actually, when it gets rolling along, it's actually quite a good SAXS station. However, keep in mind that we build it specially for this course once a year. It's a huge amount of work. Many things can go wrong. And the nice thing is that G1 and F1 operate the same way. So they'll have the same software. So if you learned it on one, you will know how to run the other. So they should be pretty good. And like I said, the quality was pretty good last year, real good last year. So don't feel that the one difference is that G1 has refrigerated size exclusion and chilled. We can work at 4 degrees. F1 is at room temperature. Now, sometimes people prefer to do room temperature. It's faster, that's for sure. So that's the only real difference. So it's a little older chromatograph. Let me remind you, this is a course, and so we've invited you to bring your own samples, and I know from past years that some of you have brought a lot of samples, hoping to get some real research. I see people smiling. I tried to tell people that this, we do our best to have these stations in top condition for you, but because of the huge workload and because of building this thing, they may not be in as good of condition as they are when you come for an actual research run, and we've been able to focus on one beamline. So just keep that in mind, and the priority here is learning how to do it and taking advantage of the expert advice while we're here. So for those of you who have never been to a synchrotron before, it's a huge particle. Wonderful! Thank you so much, Nala. It's a large, expensive facility to run. Your jaw would drop if I told you what it really costs per day. And we run 24 hours around the clock when it's operating. And so the usual mode, you're going to get a small slice of time here. Some of you, those of you who are on late at night, are welcome to work through the night. If you want to stay up, you can run as long as you want till morning. And that would be normally what you would do. You would come with, we do have individuals who come and pull a marathon and cat nap. But usually you want to come with 2 or 3 people at least, so you can work in shifts. You know, everyone's there to learn how to run the station, then you got your night shift and your morning shift and your evening shift. So that's normally how that's done, just so you know. So some of you are coming or have these night shifts where you're coming on a little bit late and you got lucky, and so you can run as late as you want and it's a longer schedule. So, but that's how it works at the synchrotron. I am told, I've never collected neutron data, I'm told the neutron collection is a little more relaxed and you can actually get a night's sleep there. I don't know, maybe Kushal can.
14 sample changer, holder, long exposure. Yeah, it's a different experience.
Yeah, it's a little
Somewhat
more intense here because of the flux and everything. Okay. All right. So I think I'm going to dive into the actual. I don't want to get off schedule here. So we will. So one last thing. So during the break time, I'll finalize the schedule so everyone knows when you're going to be running. Also, those of you who are doing size exclusion chromatography, some of you may have brought your own buffers. That's wonderful. Some of you may have brought your own columns. That's even better. Those who didn't, we need to make a list of buffers so that Irina can make up. We do have the ability to make buffers here, and we will try to make a minimum number of buffers that will satisfy everybody. So that's what we need to do, is make a list. And I'm a little bit negligent, and you did send me that information, and I haven't sat down to bring it all together. So we'll do that maybe during lunchtime, is make a list of the must-have buffers, and then I'll get that to our tech person. The priority, of course, are tonight's users. Okay. All right, so I'm going to start the regular lecture here, and I think I go till 9:30. Okay. So, that's the G line. This is Jesse. If you go to APS, you will get to work with Jesse. So he's moved on, and APS is going to be a much better place as a result of that. So this is my slide that I always show in every class. It's the big picture. That there are sort of three big things that people do with X-rays and biological samples. The first one that really has dominated the whole of X-ray science from the day X-rays were discovered is crystallography. Is that if you grow crystals, you have a highly ordered lattice of molecules frozen in position, and you get these lovely spots. We do this here, crystallography. You can learn to do that too. And it gives the highest resolution pictures available if you're able to get these, which is getting harder and harder. Because biologists are looking at more and more, all the easy fruits getting picked, and they're looking at more and more challenging complexes which might not be stable and so forth. So this is getting more challenging. Cryo-EM is an exciting new frontier that is also going to give people high resolution pictures. It's also a difficult thing to do, and difficult in different ways and time consuming. And so there are two other techniques: fiber diffraction, where you have molecules preferentially oriented, but they're not rigid in a lattice necessarily. This was historically extremely important and gives, I think, a tragically neglected field right now. And they're actually being in the field of muscle diffraction. There's some very, very exciting stuff happening at APS. And so I hope that there will be at some point a renaissance in fiber diffraction when some of the software tools begin to filter in. But at the moment, it's not a heavily used area. And then, of course, if you shine X-rays on a drop of liquid, they're completely disordered. And you get a pattern on your detector, a scattering pattern. See, here's this black square is where the direct beam comes. We protect it with something called a beam stop so the detector isn't damaged. And then you kind of see this nothing really there. And so from a crystallographer or fiber diffraction person, you would throw this data out and say that there's nothing useful. And the amazing thing is that, and as you will see, that there is a huge amount of information contained in this picture, very valuable information that you can get about the solution state. You don't need to have a rigid crystal. You don't need to have it frozen or fixed. It actually tells you, like NMR, it tells you about the behavior of your molecule under solution, possibly realistic physiological solution conditions. And so for that reason, it has happily lived side by side with higher resolution techniques, and it will continue to be very complementary. It's something that you don't, because you can shine X-rays on almost anything liquid, the possibilities are virtually unlimited. Well, okay, there's some limits we'll talk about. But so now, I like to think of this as a sunrise, or I say for graduate students who are late risers and late workers, a sunset, right? So you can think of this as the direct, as the sun is rising, this just below the horizon, and you see this beautiful color gradient going off into the darkness. And that literally is small angle scattering of visible light. And so that's the piece of data that we're looking for here, okay? So the experiment's really very simple and very, very versatile. You have some slits. You'll see all this stuff in the beamline. You have a sample, and then you're looking at, in crystallography, you have the detector up close, and the beam stop is right up by the sample almost. In SAXS, you've got this long pipe, and you have the beam stop clear at the end of the pipe, and you're looking at this very narrow, stuff that would normally be hidden for any of you who are, how many crystallographers out there right now? Okay, again, about half. Okay, this is good. So we're reaching non-crystallography audiences, which is great. So for the crystallographers, most of what you do in SAXS is hidden under the beam stop of the crystallography experiment. So it's this very, very small angle stuff. And in some ways, crystallographers talk about how wide you can go, right? That's the resolution, right? The wider out you can go in the diffraction pattern, we say, oh, that's a 3 angstrom, oh, that's a 2 angstrom, or a 1 angstrom, right? Is giving you the details of the electron density. The SAXS people are how close you can get to the beam stop. That's a 900. That's a. And you'll see why, because getting close to the beam stop is what limits how large of an object you can look at, how big of a shape you can reconstruct. So it's almost the reverse thinking of crystallography. We're trying to, and that's something to pay attention to. You may be limited, the size of your object is limited by how close you get to the beam stop and how small of an angle you can measure well. Okay. So there's plenty of time today for this series. So I just want to talk a little bit about, I love the history of this, and this article down here is fun to read. 1969 Physics Today, right? The physics magazine that you get if you're a member of the society. And there was this fellow, Andre Vignier, was a young man in the 1930s, and his professor went to him, was interested in diffraction, of course, because everyone was interested in crystal diffraction, and said, we've noticed in our crystal diffraction patterns that around the spots, in the background, there's this funny, hazy patterns that must mean something. It's called diffuse scattering. Go investigate it. Learn, tell us what this is. That was his task. And so, of course, the very first thing he did was he thought, well, I need to perfect, if I'm going to understand this haze, the one spot, diffraction spot that I should look at first would be, those of you who are crystallographers, right, your HKL index. Which HKL would that be? So it would be the one spot that every crystal, no matter how bad, has. Right? It's the one that's hiding under the beam stop, right? And so he looked really close to the beam stop, and he noticed this is an example of glass, which is silicon dioxide, and this is silica gel, which you have probably seen in the laboratory. It's used as desiccant and for chromatography experiments. And he noticed this, and it's the same composition, the same matter. But what the heck is it about silica gel that makes this strong scatter near the beam stop that you don't see with straight silicon dioxide? Anybody venture a guess? No physicists in the audience. So silica gel is a gel, and so it's got lots of cavities in it that contain water or whatever air. So they're basically whereas glass is very amorphous. What they say, it's dense. There are no cavities. It's just uniform, boring material. We're too close to boring. So it had something to do with the electron density differences, the fact that there were some molecules here and then some cavities here with no electron density. And you can look up this thesis. And this article is really amazing because at the height, he was at the pinnacle of his career as an authority, a world authority in this figure. This is his doctoral thesis, was published, and then by this time, he was the grand old man. And in this article, he writes, in 1969, he says, "We have pretty much solved all of the problems that there are to solve in SAXS. There's not much left to do," he said, theoretically. However, there's one scenario, and that is if we could get X-rays 100 times brighter, but I don't see how that's going to happen. And this was literally the eve of the first synchrotron light. And it's just that crystallographers didn't know about it yet. This was a physics experiment, and these physicists had this annoying light that they were losing energy as their particle physics, as their particles went around corners. And so ultimately, X-rays did increase by many, many orders of magnitude with modern synchrotrons, and it has led to a revolution in SAXS and the realization that there is far more to be learned. And I think that's a terribly important message to learn about science in general. Over the long haul, you know, in the short run, it may look like it's kind of worked out, but in the long haul, amazing things can happen, and we see that over time. So this is an exercise that every one of you should do, and that is go through the literature, go to PubMed, and type in SAXS and protein, and come up with all the articles from this past year, which people have done SAXS or BioSAXS. This is a smattering from a few years back that I pulled out. And what I did was I went through and I just said, what are they doing? How are they using it? This list changes. It's changing rapidly. So it's already way outdated. And I highlighted in red the stuff that is, these are the first experiments that you can do. They're easy experiments. You could use the data from this course to do these. So if you are wondering what the physiological oligomeric state is, and you have some crystal structures, you see neighbors in the crystal lattice, and you're wondering, is it a monomer? Is it a dimer? Is it this tetramer or that tetramer? And so this is an experiment that SAXS can often answer unambiguously. You put your liquid in, you do an experiment, you compare the scattering curve that you get with the theoretical curves for the different candidates. It's really simple to do. You will learn how to do that in a matter of minutes. So very powerful. You can evaluate proposed models of complex. Is it parallel? Is it end-to-end? Is it T-shaped? So you're trying to get an idea of how is this extended? Is it compact or is it disordered? Those are questions that SAXS can answer for you. So that's the wonderful thing about SAXS, is that it doesn't require that you have a folded protein. So RNA people love SAXS. It's telling them about structures. You can evaluate whether it's something is folded or unfolded or flexible. And then conformational changes. There are lots of NMR and SAXS are two solution methods that are highly complementary. We have a lot of NMR people coming to the beamline now and combining SAXS with NMR. So that's a very powerful combination right there. Building complexes from unbound components is something that sometimes it works great and sometimes it doesn't. And sometimes it's really obvious that what the missing component is and how it fits in, and sometimes it's quite ambiguous. So this is sort of an easier said than done, but it's worth a try, because sometimes you'll look at it and wow, you can see right away how things are. You may have component parts which you know, but you don't know how they fit together. And so it's kind of luck of the draw. And we will tell you later, sometimes SAXS can't tell you very much about a structure. Sometimes you just get unlucky. Certain shapes are not well suited for SAXS, and certain ones are. So I advise you to go through the literature yourself and see how people have used it. We had a case, I just loved it. We had a student who'd never done SAXS before, but who was building a crystal structure and trying to trace the chain. And as you know, if you have a structure that's not very good, and that there might be a doubt about whether or not the chain is correct. And she and her professor were not in agreement about the model. And so she ran a SAXS experiment. And when we plotted the two proposed models, it was quite clear that hers was correct. So, so this is another paper by our buddy Hayden Meritons out at the out in Petra 3, and one of the world's preeminent beam lines run by the Svergen Group. And he has this he tries to systematize the things that, these are sample characteristics that you can measure with SAXS, right? Purity is quite important. We'll spend a lot of time talking about purity. The oligomeric state, shape, which is what interests everybody, flexibility, which is of growth. I saw a statistic that like 30% or more proteins that are flexible are intrinsically disordered. I think, yeah, Kushal is nodding. That's an amazingly high number of proteins are actually disordered. Interparticle interactions is still a more physicsy thing, but there are actually pharmaceutical companies using this to develop better formulations of monoclonal antibodies and stuff like that. And conformational changes, you can see conformational changes. They have to be big enough and at the right time. These are the parameters that you get. Physical parameters. We'll show you how to do the Gideon plot. You get molecular mass and particle volume, something called radius of gyration, and a P of R function, which is a useful thing. Crystallographers will know it as the Patterson map, Patterson function, and we'll show you how to, even just getting that can tell you about conformational change. Perot exponent, you'll learn about these things that you can get. And then these are the programs that many of these are in the free but not open ASAS package, which you should try to download while you're here. And but also there are independent things like boxes, which are really great for telling you about these particular things. And so you'll learn in the tutorials of how to, the raw program can access many of these things if you download them, and it also has internal implementations of some of these algorithms. So I just want to talk about, and I'm going to stop in a few minutes and let Kushal take over, and I'll continue this after him, to talk so that you have a clear understanding of how scattering is happening and what it means to have a scattering profile. You can think of light as coming in as waves in this case, and it interacts with the electrons. So when you go to a neutron source, you have neutrons are interacting with the nuclei. It's actually the same theory. Everything you learn here pretty much will apply to neutrons if you decide to do a SANS experiment. And some areas like contrast, as you learn, are more important. So, but here we're talking about electrons interacting with light, photons. And you can think of the electrons are charged particles, and when a light wave is an oscillating electromagnetic field, it will cause the electrons to sympathetically bob like floats on an ocean. and they will make ripples that will go out in all directions. And the ripples, if you have a detector, you will see, and if you were able to do a single molecule, you would see something that's what they call a speckle pattern on your detector. And there's another way of looking at this that you might have seen, that these are the crests and troughs of the waves coming in, and when they diffract off of the electrons, they have a shift. That shift can add and subtract, and that's why you get speckle patterns of light and dark. Okay. That's diffraction. Okay. And so we're looking at this angle tells you about the shift between the waves. Okay. But in reality, things are in solution, are rolling around in disordered in their orientation, and so this pattern is smeared. And so what you see is sort of a bright, this is your sunrise right here, where the sun is, okay? So, modern life. You walk around, I'm a person who thinks out loud, and now I hear everybody talking to themselves in the store and on their cell phone, and I don't feel self-conscious anymore. I'm just on a cell phone, never mind. So this is the experiment. You're shining, right? You got this. The direct beam hits, we block it with a beam stop. We're looking at the sunrise. And so if you were to convert this into a single curve, right, of intensity, it would start at wide angle and get brighter and brighter. And you can turn this sideways, right, so that Q is your x-axis and intensity is your y, and that's the scattering profile. So that's the fundamental piece of data that you'll be collecting here at the synchrotron. Probably thousands and thousands of these if you're doing size exclusion, right? Okay, and so we talk about this variable. I've been calling this angle, but actually you'll see something called Q most of the time. And Q is really proportional to angle. It's called momentum transfer, right? It has to do with physics of colliding, momentum, billiards kinds of colliding things. And it's just almost everybody you'll see is 4 pi sine theta, but over lambda. So it's independent of wavelength. When you look at this, it doesn't matter what radiation, or even it might be neutrons, you're going to see intensity as a function of Q. It's the same Q, okay? So the wavelength is divided out. But this is basically proportional to angles. You can think of it as an angle. Yeah, here it is. Here's a little derivation of, but I just wanted to point out, when you look in the literature, here's that curve, right? You will see different variables for that angle. Sometimes, in the old literature, H, because that's crystallography, HKL, I think. You will see S in some of the Spergen programs you use. You will see S. They mean Q, and Q is what people complain about it, but Q is what we all chose. Yes.
So it's funny. I just got publication in Cellulose, and they gave me a hard time because I was calling it Q angle, and they wanted scattering vector. So they made me change all of those.
I know who that reviewer is, and he's wrong. He's very good, but he's wrong. Yeah, if you leave out a factor of, do I have it here? Yeah. Sometimes you'll see S equal to this, and that is someone who's crystallography background like this, because the inverse of that is the d-spacing, and so you can think about. But yeah, so just so you know, the world went with Q. Whoops. Okay. Hey, I think it's time for Tushal stuff. Hey, are there any questions before we go on? I haven't heard from the online audience. Oh, sorry. Question?
Yeah, so how can you get rid of this secondary scattering? Because there will be secondary scattering, right?
Oh, you're talking about multiple scattering events? In other words, a photon that scatters twice?
Electron scattering, but that will be, there will be multiple secondary scattering, not the principal scattering or the main scattering. Because when a photon is going through a molecule,
yes,
So there will be first scatter, and that scattered light will hit to another electron that
Yes.
can generate secondary scattering.
Yes, so secondary scattering is, so the probability of a photon scattering off an electron is 10 to the minus 6 at these energies. So yes, there is secondary scattering. No, you will never see it. Unlike, now, unless, of course, you have a very dense material
Yeah,
and
I mean,
perhaps
in
very
crystal.
low. Huh?
In crystal, then there will be very dense material, right? Then
In a crystal.
yeah.
Well, maybe some of the crystallographers. So multiple scattering events are important in electron diffraction, right? Because the attenuation, because the interaction of electrons with matter is very strong. The interaction of photons with matter is extremely weak. So the percentage of, so if you have a secondary event, the probability of a secondary event is extremely small, almost zero. Does that make sense? Yeah.
So question about you said about the fiber. Can we do that here, please? 'Cause
So occasionally we have set up fiber diffraction where it's not something that we do here. So I recommend you go to APS and talk with Jesse about fiber diffraction. Yeah. Okay. Are there any online comments? Questions? I'm a little concerned. I haven't heard your online people talk.
I'm in the chat, so.
Okay, so I assume that they hear us. Somebody say yes. Okay.
I don't think they're.
Oh, okay. All right. All right. So my pleasure to introduce Kushal Gupta, a longtime collaborators, and he is here from University of Pennsylvania Medical School, and we'll tell you about one of the most important things that you should know going into a synchrotron experiment: how to make your samples.
Thanks. So I've been doing small angle scattering at X-ray neutron sources. My first one was done here at Cornell, I think in 2006 or 2007. And what I've learned from, I think, dozens on dozens of trips to synchrotrons and general research reactors here.
Oh, you got a pointer.
Sorry, I got it.
Sorry.
Okay. What I've learned over the years from these trips and these many different experiments and collaborations is that it's very different from X-ray crystallography. So a number of you are crystallographers. So in the crystallography community, we have this habit of shooting first and asking questions later. Right? And so if you walk away with any other message from the next 30 minutes, it's the exact opposite with solution biophysics and especially small angle scattering. Okay. I'm going to hopefully explain why that is here. All right, and so just up front, there's some suggested readings I would highly recommend, among many others in literature now in this community, especially this most recent one here from a Diffraction group, specifically speaking to this matter. Okay, I think everyone you ask in the community about this question of sample preparation give you different shades of the same grade, basically. All right, and so these are worth looking at. And so just to review, this is the experiment that we're here about, we're very interested in, where we're taking a beam and we're taking our sample. This is probably the most important part right here. For all of this to happen, all this magic, right? We need to have a very intimate understanding of our sampling. Because if you don't, it becomes very difficult to interpret the features of the resulting profiles, shape distribution functions, and these shapes you can derive from this experimental data. This is a very low resolution approach, as will be discussed. Right? So you can't do de novo structure determination, for example, and it is challenging to directly determine some aspects of the data without a priori knowledge. So combining this analysis with other complementary methods and quality control and instrumental analyses is imperative to a successful study. The analogy I really like in terms of kind of contrasting all of this with other structural biology techniques that are at the forefront right now is the fish analogy. And so as biologists, we want to understand how the fish swims in the ocean in its native ecosystem. So our first impulse is to remove the fish from that environment and put it into a test tube. Okay. And we wanted to study it and understand it in isolation. And so the gold standard for structural biology remains X-ray crystallography. We can get high resolution atomic insights into the structure on the most basic level, right? But this experiment comes with a proviso, with a caveat, right? And that you're taking the fish and you're packing it into a sardine can in an artificial environment, all right, in an ordered way. And so while you might understand the highest resolution aspects of the scales in the fish, all right, it doesn't necessarily represent how it swims in the ocean. All right, so there's a caveat there. All right, NMR is much more closer to a solution experiment of all the techniques here listed, all right, in terms of high resolution and solution environment. But right now, even though there's a lot of innovations coming there or pushing these boundaries, you're kind of limited to small fish in a very crowded fish tank. Right. But now, as Richard had mentioned before, small scattering is being married with NMR and other structural biology approaches to enhance the information from the experiment. All right. X-ray crystal, cryo-EM. All right. The revolution right now is phenomenal. All right, in terms of the enhancements in resolution. All right, and just the explosion of very difficult systems like membrane proteins and large macromolecular assemblies and their structure determination. Right. But again, there's a proviso there. Right. You're taking a sample, you're putting it on a grid in a very unusual environment, you're vitrifying it or you're staining it, all right? So again, that really isn't a solution environment. You have to be careful. Now, in contrast to all three of these methods, which are de novo structure determination, small angle scattering is much more modest in resolution, all right? You can't do de novo structure determination because it is possible to have different shapes that give you the same scattering profile to an extent, all right? However, right, you have a lot of control over the fish tank, right? You're not limited to the size of the fish you can look at. Right? People have used scattering to look at virion particles, right? Larger assemblies, all the way down to I've done 8 kilodalton Tudor domains proteins, right? You have a lot of versatility there. You have a lot of control over the buffer environment, the fish tank contents, all right? And so there's a lot of power here, and by itself, it's not nearly as powerful as when it's combined with these other techniques. So there's some thinking, just sort of framework thinking with this. All right. Now, what's in the fish tank, right? What is our fish tank? So generally speaking, if you're working with things that are like clamshell, very discrete, very well ordered, all right, it's very straightforward, to move from the resulting scattering of this to the form factor, that is the shape information, all right? So lysozyme, MBP, cytochrome C, glucose isomerase, these are nice, very rigid, very stable species, right? Very discrete, all right? So this is a relatively straightforward process to go from the resulting scattering to a shape. Right? It gets complicated. This is where having a priori knowledge is very important. All right? But what if? And the reality is for most macromolecules, all right, that we're studying, including the intrinsically disordered proteins, intrinsically flexible proteins, okay, higher order macromolecular assemblies, you don't get clamshells a lot. The most interesting questions are things that sample a number of discrete conformers in solution at one time, all right? Or the jellyfish, extreme intrinsic disorder flexibility, all right? Then it becomes much more complicated to take the resulting scattering and transforming it into a structural understanding of what's going on the structural level, because you have a volume-weighted summation of volume-weighted contributions, all right, of all those independent species in the fish tank, all right? So understanding the aspects of your sample going into the beam is imperative to the interpretation. Okay, so it's not going to be taken lightly. All right, and then, right, and this is where we lead into now sample preparation. Impurities, all right, homogeneity of your sample. All right, oligomerization. All right, you have mixed with monomer and dimers in your sample. All right, all of these things also affect your ability to interpret the data. So if you don't have an intimate understanding of how pure your sample is, right, its concentration-dependent properties, how it behaves in different buffer environments, right, tendency to aggregate. I like to think of aggregation like cholesterol, something that's there, it's killing you, but you don't see it, okay? This will stymie your experiment. All right, so everything moving forward here are about different ways you can get at this. And this one last thing here, I'll tell you about a grad student down the hall from me, who's home graduation, now volunteer postdoc, Elizabeth Sweeney. All right, she works on a big 650 kilodalton protein called HSP104. All right, it's a big hexamer, all right? And so she had tried to crystallize this thing. It's still recalcitrant crystallization, but she was very interested in the things I was doing with small angle scattering, so she came along for a trip. She collected some data, and then she ran down the hall and said, look at this shape. All right. And then I gave her a big bucket of cold water. Right? After I asked her really hard questions, how do you know? right? Your sample is pure enough. Was it pure enough, right? Was it monodisperse? Is it only a hexamer in solution? Or is it a hexamer, trimer, mono? At some concentrations it is. Okay. And so then she made this design for a t-shirt. We promised we'd make at some point. All right, so start doing other complementary measures. Analytical centrifugation, a single species. Sec balls, right? Single peak with mass profiles across the peak, right? That are consistent with that hexamer species. So we helped validate, right, the samples that were going into the X-ray beam. And ultimately, she had a very nice publication and study about this in different nucleotide states. All right, so more important, before you pick up a pipette, right, stop and think. This is what the crystallographers fail in this regard. They shoot first, ask questions later. Stop and think. All right. Ultimately, the outcome you're looking for is reconciling a scattering profile with some model. All right. So you have the model independent analysis, things like RG and Dmax and Crowd exponent, and then there's the model dependent analyses. Right. And so ultimately, if your goal is to reconcile a scattering profile with a atomic model, right, then you have to make sure you're comparing apples to apples. All right. All too often, crystallographers have truncations, right, in their freezer, right, and they take them out, they put them in the beam, and say huzzah, right? Or they have the full-length protein, but you only have structures of truncations, things that are missing the flexible base, or vice versa, or the other way around. They might have cloning artifacts, his tags, an extra little glycine after a whole point cleavage. All right. If you really want to be successful in reconciling solution scattering experiments, atomic structures, I'll talk more about this later, you have to make sure you're comparing apples to apples. So that means thinking about the constructs going into the beam that you're making and purifying, right? What's the missing inventory? What are those missing amino acids? Or what else is on this protein that has to be accounted for? All right? Is it predicted disorder, all right, in certain regions? And how do you account for that in your modeling and analysis? All right? So there are cofactors. Is the protein stable, soluble? All right? Are you going to have enough material to use this experiment? Think about this. All right? Does it have post-translational modifications? For example, glycosylation. Right? It's a thing if you're isolating protein or from eukaryotic cells. Right? How do you account for that in your scattering experiment? This is an important consideration. Right? Are the disulfides that will be lost in the intense X-ray beam? All right? Do we have to use a different, like a home source or neutrons instead? The fusion partner, cloning artifacts. All right? Are these things going to be accounted for in your analysis? And can you make milligram quantities of this? All right? A lot of great experiments and concepts are stymied just by not having enough material. All right? So these are things you have to think about ahead of time. All right?
Hey, Kushal,
Yeah.
so I occasionally encounter people that say, oh, if you make some, you can always make more.
Yeah. So, I mean,
Radiation. So what would you say? I mean, it seems like things burn up due to radiation damage, but there are always samples, it seems, that are limited. They're just too hard and too expensive to make. And,
right.
I mean, I don't know if you
And
have
so-
something to say about that.
yeah, and so I've had a lot of projects like that. I'll tell you about one a little bit later. It's Group 2 Intron, where I was getting only 10 micrograms at a time. And I was convinced that there was no way this was going to happen. It ended up happening. But things like the sample requirements to make a static cell measurement, or the requirements to do an injection on a sec column in line with SAXS. All right. And then thinking, okay, well, and I always take the number right now, I need to do an experiment and multiply it times 3 or 4, simply because I know I'm going to have to do it again, or something's going to be an offering to the gods before I actually get success. And so a lot of it's going to be depending on that sample environment, the sample. So that's where having a conversation with your beamline contact and the scientists there, and making sure that you understand every beamline is a little bit different. Right? Every experiment's a little bit different from location to location, whether it be a home source, a neutron scattering, X-ray scattering. So having those conversations, understanding precisely what your sample requirements could be for your goals is very important, because that's going to vary from location to location. If the sample being oscillated back and forth, or you get sample, recover the sample afterwards. If it's neutron scattering, how big of a column are you going to use? And how much is going to be diluted in the SAXS format? Right? And these are all considerations to make, and how much material you really need. Be honest with yourself. We tend to be optimistic, but it can end up hurting you on the back end of the study. All right. Can small angle scattering answer the question you want to answer? Right. This is the wonderful thing about solution biophysics. You can take a model, you can make predictions, before you even pick up the pipette. All right. I'll give you an example here. Here's recombinase. All right, this protein does site-specific recombination that we studied Craig Van Dyne's group. And here's the available crystal structure of 2 monomers on a bent piece of DNA. All right. But I have a particular mutant that I think doesn't do its business because it's not bending the DNA. Right. I had to ask the question, well, will small angle scattering answer this question of resolving these 2 different models in solution? Right? So you can take these atomic models, back calculate an experimental profile, right? Back calculate a shape distribution function and see if there's actually measurable differences. There are calculators like on the MacCHESS website that additionally, in addition to back calculating the profile, will actually model an experimental noise based on the experimental configuration. Right? That's useful as well. Some sources may not give you great signal to noise, but others will. All right, so that's important. So that's the advantage of synchrotron X-ray scattering. Is that you have a very high signal to noise in the profiles you get. So you can really go after finer features of your structure. Whereas with home source X-rays, neutron scattering, a lot more noise out in the wider angle regime. It's more difficult to resolve some of these finer features in the model. All right. This is contrast variation. I'll talk more about this. This is called a Sturman plot. And in this case here, these models are readily resolved by small neutron scattering as well, just on paper. All right, so this is an example where I could convince myself I could resolve these 2 species. That was the hypothesis I was pursuing. Okay, this is very useful, right? In creating models, not only make sure you can do the experiment for beam time proposals. All right, I read a lot of beam time proposals for X-rays and neutrons. The best ones are built around a hypothesis. This is the hypothesis. This is the experiment we're going to do. This is how we expect to analyze the data. All right. Sometimes crystallographers just say, "I have crystals." Right. So if you're writing proposals for the beam lines, wherever they are, and a lot of beam lines are oversubscribed, so they're competitive, right? Doing this is a very worthwhile activity, even if you don't have preliminary data just yet.
This is an important point to make. Years ago, when I didn't start out in crystallography, I got into it almost 20 years ago. And, prior to that time, if you just published an X-ray structure, you could publish it. It would make the news. But right about the time I got in, the biologists on campus were saying, well, you can't just publish a paper based on one structure anymore. It's got to be part of a bigger picture. And SAXS has, from day one, I think, has always been in that position. You can't really build a whole paper around just SAXS data. It's got to be, it has to be a part of a bigger picture with other biophysical methods. It just doesn't, it isn't going to get, even if it gave you a full structure, you can't just publish a structure anymore.
So I like to think of it as sort of like duct tape or glue. It can really pull together different types of data in a very elegant way. Right? So it's very useful in that regard. Along the same vein, solution biophysics in general, where you're doing light scattering, right, AUC, you can simulate data from first principles, right? And then just ask yourself the question, can I see the changes I'm looking for? Right? Do that exercise. There's a lot of great software out there. I'm not going to go into detail here that can help you do that. All right. So it's worthwhile just doing these calculations, making sure the experiment is worth your while and the investment material, especially working with eukaryotic protein and precious amounts of sample or drugs or whatever it may be that reagent you need for the experiment and the man hours. Right? So it's very important to make these calculations up front and convince yourself you do that. So for example, I was able to model for sedimentation velocity, what a peak would look like sedimentation velocity with different frictional coefficients, different shapes, right? And I convinced myself that the result was not only meaningful, but was consistent with a particular elongated shape of the solution. All right, sedimentation equilibrium analysis, you can do this. Basically, we can model, the slope of these lines here. This is natural log versus radial position in a sedimentation experiment. The slope of the lines is proportional to the mass, right? And so you can convince yourself, okay, I can resolve dimer, tetramer, octamer, and experimentally I see some instances of tetramer, for example. Right? These types of comparisons and calculations are very important. Not only in communicating to your readers and the proposal readers and the grant readers, but also to convince yourself that your interpretation is what you think it is, not some alternative possibility. All right. I'm going through it at a brisk pace here. I want to make sure we keep us on time here. But if you have any questions, please just jump in. Okay. How much macromolecule do I need for this experiment? All right. And so you're going to hear about this master equation more in a bit. All right. And so the resulting scattering as a function of scattering angle is proportional to the volume of the particle, the concentration of the number of particles per unit volume. Right. This contrast term, which we'll talk more about later, the form factor, right, and the structure factor, which is hopefully negligible in the experiment with regards to deriving shape information. That'll be discussed more too. But really, the short of it is that the intensity of scattering that you get from your experiment, right, whatever source it may be, is proportional to the concentration of the mass. Right? So you have enough macromolecules, enough solute in your solution to get a clean signal. Okay, and so generally, best practice, you do a series of concentrations, right, and see and kind of probe a range of concentrations, right, in your experiment, right, making sure that RG is stable and that things move in a predictable and way with regards to your understanding of the oligomeric state of the macromolecule size and shape. So generally in practice, you do this, look at the Guinea regions, right, to make sure there's no aggregation or interparticle interference, right? And this ends up being in practice for most macromolecules, most preparations, in a regime of 1 to 10 mg per mil, all right? But a good rule of thumb to start off with that people like to use is that you take the molecular mass and KD, multiply their concentration mg per mil, should be somewhere near 100 to start. Need probe above and below. That's a good place to start for most experiments. All right. Talk to the scientists at the different beam lines that you'll be using, all right, because different setups have different requirements to sample. Some are down to 30 to 20. Some are up to 80 to 100, depending if it's a capillary or if it's a plate robot or if it's, any of a number of ways the sample is being delivered to the beam. Okay. Sample requirements might be much larger for a BioSAXS experiment if you're injecting into a larger column that's going to be diluted through that column process. Right, might be injecting 100 microliter bolus or 300 microliter bolus. It's going to depend on the column you're using in that case. All right, small neutron scattering is an expensive experiment that you're using a lot of material. It's a relatively weak source of neutrons. You use banjo cuvettes, these little cuvettes that are used for CD, and anywhere between 350 to 700 microliters for a given sample in solution. The exposures are much longer, but you're also doing multiple contrast points in addition to the concentration. So the neutron experiment is a bigger investment in material than synchrotron SAXS side by side.
Kushal, can you reuse the material that's been in the neutron beam?
Yes, that's the neat thing, is that you can recover the material. Unlike X-rays, neutrons are non-destructive. Right? You'll not have radiation damage at the sample, although you may have activation of elements from the neutrons that make it radioactive. Right? So there's a cool down period after the sample's been exposed, depending on what's in the buffer. And you can recover it, do protein assays on it, or test for activity afterwards as well. There is a recovery you can do in a neutron experiment, depending on the radioactivity calculation, the activation calculation.
So, in principle, you can recover sample from a SAXS experiment, and probably as a percentage, there's not a lot of damage, but you have to understand the sample has been damaged and contaminated and would need to be repurified.
Right.
So, and it's oftentimes a little awkward. It's easier now to recover. You can recover sample, but very few people do it.
So generally for any sample, these are the questions among many to ask, right? If it's suitable for a scattering experiment. Is it chemically monodispersed? Is it physically monodispersed? Is it sampling many conformations at one time, or is it the discrete clamshell versus the jellyfish? All right. Stability, is it happy in the buffers you can be using? If you freeze and thaw it after you bring it to the beam line, will it still be happy in its solution? Will it be aggregate free, or do you need to size it again? All right. Understanding its concentration dependent behavior and buffer behavior, storage shelf life, right? All those things matter in being successful in your experiment. All right. Concentration dependent behavior, oligomerization, aggregation, interparticle interactions. If you're working with nucleic acids, interparticle interactions are a major consideration. All right. For example, nucleosomes is a good example. It's a big ring of DNA, 50% by mass on the outside. Buffer conditions are and concentration conditions are very important to avoid interparticle interactions. Okay. Unhappy fish, bad. Right. All right. Understanding the concentration of your sample. We tend to be cavalier about this in most applications. We're okay with the bracket being off by 10 to 15% most of the time. However, if you don't have a good way of assessing the concentration of your sample, right, it could hurt your experiment later. All right. How do you know that the nanodrop reading for a nucleosome is actually what it is? I had this conversation with this past week. right? Is a nanodrop reading on your particle the same as a Bradford reading? And which one is correct? All right. What's the wider error? How do you figure that out? Okay. Because those in neutron scattering, you can have the opportunity and necessity to directly reconcile the mass and the concentration of your particle with the I of zero information. Right? It's very compositionally sensitive. If you don't have a good concentration determination, you're really undermining a lot of your analyses. Okay. And so generally, these are things to think about. Some classic references here in terms of using extinction coefficients theoretically and how to experimentally determine them. Sometimes the data bench is worth this step here if you have the material to spare. All right. Getting a hyperchromic correction factor for nucleic acids is usually very useful as well. Sort of a fudge factor from the absorbance reading of a DNA before and after digestion with an enzyme. Okay. Those things can help you improve the accuracy of absorbance reading relating to concentration. All right. Bradfords, be very careful because they can be off by a lot if you don't do it very carefully. Even if you do it carefully, it can be off by a lot. All right. So do multiple concentrations, work within your assay range. Don't use old Bradford reagent. Don't use old standard curves. All right. Do it fresh, right? And do it right. And then you can take that information and do complementary measures from your primary data. I of zero from SAXS. If you have good concentrations and a good standard, right, derive mass one of several ways, right? That information helps you later, right? You can use a refractive index from your SEC-MOLs run and see and just kind of do a reality check of the concentration of your macromolecule, right? And also FCIR has some applications. I think there's a little box that was in the wet lab at one point used to determine.
Yeah, it's one of those things I keep wishing that more people would use, but we have had, for some people with either detergents or lipids, I think really love the device.
Yeah.
There is, I've heard people using index of refraction.
Yes.
And I wonder,
Very good.
the RI has it, but as a regular means of measuring concentration, and I wonder, it sounds really like a great idea.
It is because, irrespective of sequence, right? Proteins always have a DNDC of 0.185, right? And so that's why SEC-MOLs leans on it heavily to get a very precise determination of concentration as your molecule loops off with a gel filtration column to get that light scattering information. So there's other methods aside from just Bradford and nanodrops available to you to determine concentration very precisely. Right? So it's definitely worth the time to check that. All right, purity. Right. So generally speaking, crystallographers are in a good place there because they understand the importance of purity, 95 and 98% pure on STS page gel. Looking out for things like DNA Ks, high order Schmutz, even off of the gel filtration column. This was, for me, concerning in this study, even though it was really pure. This is the prototype virus integrase, right? These little proteolysis bands, so they can contribute to your scatter, right? So really being very, very fussy about this. I've sent collaborators back asking them to do silver stain gels, right? Just to illuminate exactly how pure this stuff is after just one nickel column. And that usually is quite illuminating, right? Generally speaking, I usually advise at least three purification steps, including sizing at the end, right? Ion exchange is great because it gets rid of the things you can't see, the proteases from bacteria and so forth. And then a good, affinity purification step at the onset to get it to 95% pure at that stage.
So this might be a good time to bring up the students. In general, you have two choices going to a SAXS line. You can do conventional, I'm not sure how to call it anymore. Some people say equilibrium SAXS, or where you just have a sample that you might oscillate in the beam. Robotic SAXS is what we call it here. But you also have the option of using a direct size exclusion or a chromatograph directly connected. Now, some people argue that you should just always do that because you never know. There's always some differences in things in your peaks, but I tend to think that you should choose carefully depending. What's your opinion on that?
So to restate the question, the opinion on
size exclusion or regular robotic SAXS, which how do you make that decision?
I think a lot of it comes from your understanding of the sample, right? So for example, I study HIV integrase, which is this miserable label. All right. SAG cell measurements aren't very useful. And I can show you an example of that in a bit and explain why that is. Whereas SEXAX is very, very powerful, right? Way of analyzing that equilibrium, those mixtures, right? So even in the gel filtration format. A lot of it depends on your understanding of the sample and how discrete it is. All right. If your sample is prone to aggregation, a sizing step immediately before the measurement is outstanding. You're going to get rid of those aggregates in the very last second before it goes in front of the beam. Right? So the quality of data goes up quite a bit in terms of beginning analyses and so forth, and getting precise masses. Right? I generally now favor the SEXAX approach to getting the best data possible. Although I think that obviously both ways are very useful. But in terms of data quality and getting rid of these considerations, these things you can't see, and these other problems that could be in some more challenging systems, the SEXAX is great. You're getting rid of the aggregates at the very last second, right? Not letting it percolate and sit around to gather more. And with the new computational methods available, you can start resolving what's going on in that peak.
So the robotic SAX, I think, does have the advantage that you can use a more concentrated sample and you can get a better signal because of that. And also, I think if you're going to do titrations or something like that, you can't really do those with the size exclusion. Size exclusion introduces dilution. So I think we give people the choice. You can decide on the spot with the push of a button which one you want to use. But I think also you want to be careful, like as Kushal is telling you, it pays to, don't depend on coming to the synchrotron and the size exclusion solving all of your purification problems. I think you probably agree with. It's better to solve those first.
It might heal the sick, but it won't raise the dead. That might be a good way to say. Be a good biochemist, and take heed of some of these warnings here with regards, especially working with vendor. I'm sorry, you had a question.
Just furthering his question, are there really any advantages to not doing the SEC other than precisely being able to control the concentration and also using less sample?
If you have, so less sample is a big one, I think, right?
But are there any other advantages besides
if you need precise control of concentration, right, then I always call it static cell, but the fixed cell format is very useful. If you worry about dilution effects, if part of your analysis is resting on relating the scattering profiles to concentration, right, and some concentration-dependent behavior, then it may be difficult. It may be a moving target with regards to the size exclusion, right? So there are definitely advantages.
It also is a slow experiment. For a single injection, even at room temperature, the best I've heard is that you could do, 3 samples in an hour. Whereas in reality, and that's, if you really want to resolve something and you need to keep it refrigerated, your size exclusion experiment could run an hour for one sample.
Whereas the throughput in the other method is much higher.
But if you have time and protein, you're not worried about the concentration as much. There are no drawbacks to doing SEC, and you get significantly more.
Yeah, I mean, if you have the time, if you have the sample, if you're not trying to change buffers a lot, if you don't have a lot of ligands you want to titrate in, if you don't want to try a bunch of different buffer conditions, given all that, yeah, SEC is beautiful. But if any of those don't really fit your experiment, then it may not be. I think there are advantages to both, for sure.
I'll just chime in as well. I'll show an experiment we did in my talk this afternoon, where we used both techniques, and both were necessary for answering questions about the same protein. So, so there are cases where one is preferred.
All right. Keep the pace going. I want to make sure we don't run over.
I'm looking at the
too much. And so I direct a structural biology and biophysics course at Penn, which we lean on a lot of instrumental analyses. All right, and I take advantage of them to make sure that the scattering experiments that we do are successful. So there are many different ways, all right, many techniques out there, all right, that you can use to help inform your knowledge of the sample you're putting in the beam. Do you have to do all of them? No, all right, but doing a couple, at least 1, 2, right, will help you immensely, all right, in finding success with your scattering experiment. I'm going to highlight some of them here, all right, some of my favorites. Usually they're light scattering, right? Or they are anti-centrifugation. Those are my two favorites with regards to the interplay with small angle scattering. Okay, and all these different methods you have available to you at your respective institutions and locations provide you a lot of complementary information to your small angle scattering analysis. Generally, these are the figure ones of your scattering study. Or you have to convince your readers, you have to convince your reviewers that the stuff you're putting in the beam isn't an unhappy fish, isn't bad, okay? That you have to convince them that the stuff that you're putting in the beam is exactly what it is that the experiment demands, okay? I'm studying this group 2 neutron particle from Marlene Belford's group. She sent me a couple different variants. We call them delta A and plus A with regards to the catalytic adenine in this particle. And we wanted to do EM and small angle scattering on these particles, right? And so, very precious amount of material. So I wanted to use size exclusion chromatography first to just see what the behavior is. I didn't have a lot. I couldn't do a lot of different measurements in just one fell swoop. Size exclusion chromatography is the simplest and most straightforward of biophysical experiments you can do, right? Because the retention time is based on the radius of a hard sphere, directly related to that. Okay, it's not molecular mass, even though we use molecular weight standards. It's not molecular weight. It's the radius of a hard sphere as it travels through that particular medium. Okay, and so the plus A particle is the one we ended up studying extensively, right? The whole 9 yards. And the reasons are obvious, and that's a nice, well-formed, nice symmetrical peak. All right. This delta A particle is miserable. All right. Do the first injection, I get this solid red line here. All right. There's something coming at the void. All right. That's a bad sign. It's really large and distended. But the worst sign of it, aside from this the horrible peak property, is that an hour later, if I do the same injection, same sample on ice, it changed dramatically. All right. That's a bad sign. And so that never went near an X-ray beam. Right. This is the simplest of experiments you can do in an afternoon, just to say, okay, yes, yes, no. Right. This is a sample. I'll take the synchrotron. I'm not going to bother wasting the time over here. Okay. Light scattering. I love light scattering, right? Because I think it's really, just a close cousin of X-ray scattering and neutron scattering. Because a lot of the principles, the first principles are the same in terms of the angular dependent scattering and the phenomena that's occurring in terms of elastic scattering. Okay. And so generally speaking, what this means is that, two common tools you may have in your institution if you're in a biochemistry or chemistry or other related science department are dynamic light scattering and some variety of multi-angle light scattering in line with size exclusion or maybe even standalone, right, the older application. All right, these are great because you don't need crystals, right? You don't need high sample concentration to do these analyses. You can use roughly modest amounts of sample. All right, it's a very rapid reading. A student, a trainee, graduate student, postdoc, anyone can walk into an instrument and make a measurement within minutes to an hour, right, on a prepared instrument. All right, broadly applicable to all varieties of macromolecules of shapes and sizes, with the caveat that smaller things need higher concentrations to get signal. Right? Scattering power directly related to the concentration and the volume of a particle. All right, very few buffer restrictions. I think the big things I usually keep about are things like glycerol and very heavy excipients and detergents stuff. All right, but there's very few restrictions in general. The big disadvantage, obviously, of course, it's low resolution. Right, so we'll highlight some of that right now. All right, dynamic light scattering. Okay, this technique, right, very useful. Right, based on the Brownian motion of particles in solution, and then basically modeling that motion using the autocorrelation function. All right, this is very good at telling you that if you have aggregation in your sample or not. Right? But it's very horrible, not very good if you want to ask a question, do you have a monomer or a dimer? Right? Because it's very possible for an extended monomer, a compact dimer, I can cite some examples in practice, HIV integrase is a good one, where you have the same exact hydrodynamic radius. Right, so you couldn't resolve small oligomers, for example. But this technique is very quick and very easy to see. Do I have aggregation? Do I have aggregation? Do I have aggregation? Right, so very useful to use this. All right, stack mols over the past decade have become basically a workhorse of structural biology. We use it daily, all right, in quality control macromolecules we're making in different which ways. All right, and so basically this is a classic gel filtration experiment where you have some reservoir solvent, inline degasser because air scatters as a degas buffer. right? Your classic sizing column, a good separation, the analysis, whether it be SEC-SACS or SEC-mols, is only as good as the quality of the separation gained from your gel filtration column. So thinking about what column you're using here is very important. And then in line with these two boxes there, we use wide instruments at Penn. One is a 18-angle light scattering instrument, that also comes in 3-angle variety, and one is a refractive interferometer. Right? You can basically combine all that information together to derive absolute mass of your macromolecule in 1 second increments and map it across your eluent, your gel filtration peak from UV or RI, and get mass profiles. Sometimes you see a nice straight line across because this discrete clamshell species, right? Sometimes you may see mass profiles, the function of the concentration of the sample you inject. That's a bad sign. That means even within a peak, you can have oligomerization in multiple species, right? So being mindful of that, especially if you're going into a SEC-SACS experiment, is very, very useful. And so the concentration-dependent behavior, even within a peak, can be quite informative to tell you do not want to use something or have certain considerations to make in your experiment. All right. I call aggregation the silent killer, like cholesterol, right? Here's that group 2 intron particle again. By UV, looks swell, right? But if you look at the other information from the size exclusion from the SEC-mols experiment, the light scattering information. This is dynamic light scattering in line, pink here, and red is one to many angles of light scattering near 90, right? What you see here at the void from the preparation to collaborate with Centimeters, this gigantic whopping amount of aggregation. Strong scattering power that will affect your scattering experiment at the beam level, right? And so in this case here, we had to use that special filtration. You can see here how the aggregation is kind of bleeding into your peak species, right? That will kill your scattering experiment. And so we had to take special measures to basically filter the sample through a filter membrane right before the experiment, right? Just to get rid of this because we knew what was happening as a function of time and concentration. All right. So again, SEC-mols is very useful because you can go through and just go through samples and ask the question. Yes, I'll scatter this one. Mass is right, discrete species. Not going to do this one because it had aggregation leading edge during the injection. It's going to be a problem in my experiment. It could be a problem in analyses. Even they might be able to flush and work that out. All right. This one, yes. So I commonly use it as sort of just walking through, saying yes, no, right? Informing my understanding of the samples going into the scattering experiment, SEC-SACS or otherwise.
So I was so impressed by this talk when I first heard Kushal gave it many years ago. And we bought a Wyatt instrument, and we have that inline so that you can do SEC-SACS mols, not today, but when you come back someday. And mols is basically the visible light analog of SACS. And there's some differences, but the one thing that maybe Kushal can comment on this is that so for the most part, it's a great additional detector that you can hang on there so that you can see things that you may not have seen. But you can also quantitate that data. But if you want to do real quantitative measurements on your SEC-mols experiment, you have to do these instruments evidently require a very long equilibration time, and so you want to. It can take as long as 6 hours to settle down to an optimum noise level.
It depends on what type of column you're using. If you're using the GE columns like the S200, SuperDex 75, and so forth, they shed fines. Those fines basically affect your scattering background, right? Whereas if you use silica-based resins, right, the baseline is always flat and very clean on light scatter, right? And so the extension of that is that with regards to getting good baseline noise and within parameters, a good amount of equilibration time definitely is indicated.
Yeah, we're learning this from APS. Sir, if you visit APS and work with Srinivas Chakravarti, he's a real expert in the size exclusion and the MALS, and he will the MALS is an experiment that takes an extra amount of time. So if you depend on that, remember you're welcome to use it with us as just an additional detector. But if you really want to quantitate that information, be prepared for extra time in your experiments.
Be patient.
Yeah. Did we have a question?
Is
Yes.
that the 3, 8, or 18 angle?
We have I believe it is the 18. Do we have 18? It's 18. Yeah, we have a dynamic DLS. One of those channels is dynamic light scattering. And I have been told that the dynamic light scattering, it's there. We haven't made a lot of use of it. And to make a really good dynamic light scattering, you actually need multiple channels. So that particular arrangement, the DLS, is sort of a nice add-on, but I'm not I don't.
In practice, it's hard to get that data off of a really beefy sample.
Yeah.
For DLS samples, what's the smallest size you can reliably measure with that?
Question. I think after, I think it becomes very fuzzy. It's not even so much making a measurement, it's the precision of the measurement, right? Is it really 10 angstroms or is it really 7 angstroms? Is it really 10 nanometers or 7 nanometers? Or is it really 12 nanometers, right? Bigger things give you much cleaner, much more precise results, but the small things, you could have some fluctuation. It's difficult to get that data.
Like what do you like cytochrome C?
You can overcome issues with size by going to higher concentrations, but by going to higher concentrations, you actually be fueling aggregation. It's a circular problem. You keep chasing your tail, basically. Does that make sense? In that, it's going to depend a lot on the behavior of that particular macromolecule.
Basically, smaller molecules, you don't have that much reliability on their sizes.
The absolute number may not be reliable, but it will clearly tell you if there's aggregation in that sample or it's polydispersed. But very small things are challenging in that regard.
So I think the beamline out at Petra 3, I think that they insist that you do a DLS experiment before bringing your, maybe Jesse knows or not, but I remember they were strongly recommending that for static SAXS to ensure that you have monodispersity, and it was a good way to, although I've known a lot of people who have tried to use DLS machines and not gotten very good results out of them over the years. So it's another one of those things that I think you have to do right if you're going to do it.
Yeah, it's going to take some work to really get the data that you're happy with. Yeah, 10 more minutes, but I'm going to wrap it up as quickly as I can. Sorry, it's fully wrecked.
No, it's all right. My talk, one of my talks is really short, so you're welcome to take some time.
The other technique I really like in terms of categorizing samples for scattering analysis is analytic centrifugation. All right. First principle is a very old approach. Okay, where basically, depending on the rotor speeds that you're using, in the case of sedimentation velocity experiment, you're working at rotor speeds where the centrifugal force, right, is much greater. That is the force pushing the sample to the outermost extreme of the cell, the outermost radii, is in excess of the buoyant force and the diffusion force, right? So that the rate at which this particle sediments to the bottom in these conditions tells you a lot about the size and the shape of the molecule in solution. All right. And so here's an example, again, that group 2 neutron particle, right? And so you can determine things like sedimentation coefficient in a given buffer condition are compared to S20 water. The frictional coefficient, which gives you comments on the shape, 2 being very elongated, 1.2 being near spherical, okay? And there are many different robust programs now available to help you do different types of fitting of this experimental data to help you derive things like the CFS distribution. This is the fitting of the Lamb equation, the boundaries of absorbance. This is basically the radial position. This is the absorbance, right? You can see that at time points near 0, you have basically this sample, you have this boundary that starts here. And as a function of time, the boundary moves along. T plus 30 seconds, next increment, next increment. You can model these boundaries with the Lamb equation to derive what's called a CFS distribution as a description of the particles in that solution, right? It's a function of their S value. Okay. Very, very useful approach, commonly used in industry to quality control biologics. For example, it's very sensitive to aggregation. Okay. And so here you have an example of one particle that has a large frictional coefficient hand in hand with S value. All right. And then here's another particle that's a little bit more globular, right? The lower frictional coefficient, the much larger S value. S value and frictional coefficient are inversely correlated. Higher the frictional coefficient, lower the S value, and then vice versa. We'll mix that up a lot of times. right? You can have the same stoichiometry but very different shapes, give you very different S values. You can take a crystal structure, back calculate what the S value should be and the frictional coefficient should be, and compare it to your experimental data. So that's very useful in practice. Like just doing a reality check on your data and vice versa your modeling. All right. But teaching equilibrium, basically you're doing the experiment a little bit differently. You're using lower rotor speed, so that centrifugal force is exactly opposed by the diffusion of the Boyne force, right? So that after a long period of equilibration, sample will equilibrate to a certain position, radial position, based on size, independent of shape in any which way. Right. This is the natural log of absorbance, based in essence, versus radial position. So the slope of this line, in the simplest of analyses, can be related directly to a oligomeric state. Right. But this is a very simple extrapolation. It doesn't tell you about association kinetics, right, or thermodynamics in that regard. Right? You can take the data and more robustly model several different concentrations and speeds together to derive association constants and fit different models of association. Test different models of oligomerization, for example. Is it a monomer dimer? If so, what's the KD? Is it a monomer dimer tetramer? Is it a dimer tetramer? Is it a dimer tetramer octet? Right? You can walk through and test your understanding of what's going on in solution by testing these models directly and looking for the best statistical correlation. Right, so that's very useful if you have something that's there that has a concentration-dependent behavior. All right. In the end here, all right, are you scattering and mixture? And then I'm going to lay off of this simply because I think this is going to be this will be covered more later in a couple different sessions, right? But even though you might have a single peak, or the I of 0 may be constant in terms of mass, right? Calculation, you can still have something that's very complex, right? And it's actually not what you think it is. So I have this case of HIV integrase. Right? This is a protein that's monomer, dimer, or octamer. It'll be whatever you want it to be, and will aggregate, right? And even though you may do a gel filtration, you see a nice single peak. If you do different concentrations of injections, you see the mass profile of secmoles varies with concentration, right? So this is, I know from BioSAXS experiments now, this is sampling monomer, dimer, tetramer, depending on the mutant and the variation that you're using, right? And so it's never one thing, and you can do first principle calculations again, just using association constants from sedimentation equilibrium, and find that in the concentration range you may work in for in vitro analyses in the micromolar to millimolar regimes, you never have just one species. You always have a mixture, right? So that's not a great candidate for scattering unless you have other ways of getting at extrapolating, deconvoluting that information from the data. This is an example where I wouldn't just do a traditional scattering experiment. All right, so that's one case example. All right, I'll wrap it up here. Very important. All right, now you got your well understood sample, you're going to the beamline. All right, talk to your beamline contacts, talk to the scientists about your experiment, let them know that you might be doing something unusual, ask them about the requirements to do the experiment in terms of time, sample volumes, your ability to get at a wet lab space to manage your sample. Do you have a deli case? Do you have a -80? Do you have a -20? Do you have dry ice? All right, go through the whole bucket list. Don't assume. When I started going to synchrotrons for experiments, I used to pack half the lab in my car.
Just believe me.
Quite literally. I would walk in with these hand trucks of stuff because I didn't want to assume anything. But now it's much, much better. I think just generally speaking, the whole community understands the importance of a good wet lab hand in hand with the synchrotron beamline. You want to manage the samples very closely, immediately before and also maybe afterwards, right? In terms of quantitating concentration and taking samples, the things that you'll bring back to your institution. Okay. Doing dialysis. Does it have a centrifuge to do a high-speed spin to give it an accuracy immediately before your measurement? This is a very important concept. So the buffer match. On the slide, I think it's the next slide. Sorry. I'm trying to wrap it up here. All right. A good buffer match is imperative. It is so imperative. It is so imperative to getting good data. With size exclusion chromatography, that problem is solved. All right. For the most part, you separate any buffer artifacts away from your peak species, which is in the running buffer. It's beautiful in that regard. You get always great buffer matches. But if you're doing the other, sort of the static experiment or neutron scatter or home source measurement, right? Spend a lot of time getting a good buffer match. This means doing a long extended dialysis, right, of your sample, or taking the sample off the gel filtration column with a high enough concentration and using the gel filtration buffer as your match, all right? Or the last flow through from a spin column or a concentrator, right? But making sure that the match buffer, it's not just an optimal match, but a true match to your sample. Because if you can't properly subtract the solute scattering from the buffer alone scattering, right? It's extremely difficult to rescue the experiment, right? So a lot of careful consideration. Don't just try to mix it by hand and think you'll be close enough. You won't, right? Do it by dialysis, concentration, gel filtration, right? And do it properly, because otherwise you're going to hurt yourself and you're going to get results that won't make any sense, right? And then this you can bring up. Basically, there's things that shouldn't add to the buffers, right? They might affect your contrast and the signal intensity of your experiment as well. Synopsis. All right. Think before you leave. All right. It's not crystallography. All right. Use other approaches. Okay. Get that figure 1. Convince your readers and your reviewers that you have the right stuff through scattering, and learn some biophysics. All right, because it really is going to help you a lot in this regard. And I'll leave it at that. Thank you.
All of this stuff after it, or I was going to do 15 minutes of lecture. I can break that right away. Let me just get 15 minutes, and then we'll be on schedule. It did. Probably not a full meeting. Yeah, it did. Yeah. Okay. So. Maybe I should take a break. If you want a break first. Let's stay on schedule. All right. So, there we go. All right. Yeah, that HDMI looks nice. All right. Okay. This was the math page I showed you a minute ago, so let's get that. Yeah, so this is, yeah, I can't overemphasize what Kushal just said about matched buffer. And it's not something that I think people are used to thinking about. And you might do a blank in spectroscopy, but in SAXS, having the buffer exactly match what's really in your solution is so important. And most of the time, you all know these preparations are really quite a cocktail. And so it's best to change the buffer in some way. And there are, Kushal mentioned several ways. Size exclusion is a great way. Dialysis, or you can use centrifugal concentrators. Put your sample in, put thin volumes of buffer through, and then the Ando group uses these they're like mini size exclusion columns, ion exchange columns, which they swear by. So there are several ways to do that buffer exchange that you need to do. It's very important, because otherwise, you need to subtract away the water so that you can see what the protein looks like. And so here's a typical background scattering water and X-ray parasitic scatter, right? It's this baseline. And then here's your buffer. And you want to see those tails come together. If you see them cross, you're in real trouble. And this, or if you see them too wide apart, it can be real trouble. So that's very important. And as you'll see in a few minutes. So, and that can be tricky if you have highly viscous sample. That's what we've seen people doing sucrose gradients and things like that, or if you have a lot of PEG. Sometimes it's difficult to get the match. Background match. So, but extremely important. So, Kushal pointed to this equation, and I'm going to come back to this over and over. It's a very simple equation that just tells the whole story of what you need to know in SAXS. And I've actually implemented, we have a paper coming out with 2 undergraduate students that work with me. We finally published in ActiCRIS a paper where we implemented the whole 9 yards of how do you simulate if you have a theoretical profile and you want to simulate and you know the concentration and the molecular weight and all of those things, and the photons, how strong your source is. I'll show you an example of that. And it's a web application that you can use to kind of get an idea of what concentration you need and whether or not you'll be able to see what you want to see. So, but so again, your scattering profile is proportional molecular weight in kilodaltons times concentration in mgs per mil. And then there's this contrast term. We'll talk a little about this. And then there's this thing which represents the shape. It's the shape of the particle in scattering space. And then this S, which for the most part people assume is just 1 if you have a dilute solution. And so we won't really talk a lot about it, but there's very interesting things that can be done with this factor, and I'll talk a little bit about what that tells you, what that factor tells you. But this is the one that that is the one that most people are interested in. That's really what you're trying to extract from the experiment, along with the molecular mass. So, okay, yeah, so you can tell right away scattering goes as m times c, so large particles scatter much more strongly than small particles. That's why aggregation is a problem. If you've got big aggregates or precipitates, they are going to dominate your signal and drown out what you're trying to see. So that's why you want monodispersity. You want the largest particle in your system to be the thing that you're looking for, that you want to see. Small stuff, less important. Okay. Yep, okay, so we talked about that being shaped. So this is where all those the pictures of the ripples in the pond I was showing you, that's how the shape of the molecule translates into the shape of the scattering curve, the diffraction part, right? And you can write down the formula for a sphere. I just like to have equations. I'm a math major, so you can write down if you had a sphere of constant electron density. In fact, there are tables of all the different shapes. You know, the soft matter people, rods and cylinders and concentric circles and toruses and everything. People have written down the mathematical shape of this form factor, and the sphere has sort of this. It looks like a bouncing ball as a function of Q, and so if you have a small ball like lysozyme, it's a very spread out bouncy ball. And if you have a large thing like a virus core particle, it's got a lot of bounces in it, right? So that's the nature of scattering. Big things show up at small angles. Small things are really spread out. So that's the important lesson there. Yeah, so this was an actual virus core particle that somebody sent me many years ago. And this green thing is the buffer, and you can see the sample. There's your bounces. Now this is not a typical, this is a cracky plot. I keep meaning to put an ordinary plot here, but you can see it's real. It's, I mean, physics works. It's pretty cool. So this is a table, I just love this. This is a theoretical paper years ago by Volkoff and Stergan, and they took these geometric patterns, they calculated what the scattering curve should look like. Look at that, S. See, they use S. And you can kind of learn from this table. You can glance at it. See, these are the ones that are spherical. You know, 5, 6. The ones that are sort of spherical. If you have a long rod like 10, it's kind of pulled out like that, or a big disc like 12. They're kind of elongated. It's hard to tell the difference between a rod and just looking, but. This was kind of a sobering picture because these were the bead, people tried to reconstruct the shape from the profile, and these are algorithms which are available today for reconstructing shape. And you can see that sometimes it does a good job, and sometimes it's not so good, especially big disks. So this is kind of a sobering, or if you have a ring, you think you've got a ring, be careful because SAXS, these bead models, they don't do rings so well. Maybe Thomas can say, is Thomas in the room? No, he disappeared for a little bit. Okay. Yeah. I'm curious to find out, did you guys run use the DNA polymerase?
It suffers the same ambiguity problems, right? The problem is that as Richard said in his earlier lecture, you're looking at molecules in solution, so they all have different limitations. So you can imagine if you had half a ring, you're going to measure the half ring here and the half ring there and there. And you're not going to be able to tell the difference between a ring that's in those same orientations. So that's why rings are so hard to construct, and the electron density function is the same ambiguities from the scattering.
That's what I was curious about.
Because you reconstruct back against that.
Okay. So beware of ring. If you're looking for mechanisms of ring opening, be careful how you interpret SAXS data. Okay. So a couple more minutes and we'll take a break here. This is our friend Professor Guinier, Mr. Guinier, and he developed this famous formula. You can look in his thesis, and it's that the scattering is basically a Gaussian curve. Look at that. It's a Gaussian with a funny tail. And the width of the Gaussian is related to something called the radius of gyration. And the height of the Gaussian is the intensity of the curve of the scattering if you were able to look under the beam stop and you weren't blinded by the direct beam. That's what that is. So these are the two parameters that everybody gets from your first SAXS experiment. And to this day, we still get these, and I'll explain why in a minute. This I0 and RG. You do this Guinier analysis, and that he's known for. The radius of gyration, you can understand. I'll show you a visual way to understand what this is. You can imagine it's like a radius, but not quite. It's sort of, if you take the difference between the center of mass and every electron, and you sort of sum the squares. That's what, and square root, that's what it is. But the point is that the Guinier analysis, if you take the logarithm of both sides of this equation, you will see, and plot it as a function of q squared, you will see that at small angles, you will get a line. And the slope of the line will be this thing here, minus the third part. So that is the Guinier analysis. You plot your data log I as a function of q. And the raw software will do this. We'll show you how this is the very first thing that you do with your data set. And then you get the slope and you extrapolate to 0, and that gives you the I0. And that's how you perform the Guinier analysis. Now, these things are not all that useful. You can see if something changes in size. But there's another reason why we do this analysis. But here's just some examples of the radius of gyration. Because it is a sort of a sum of squares, these are different ways of thinking about it. I like to think about it this way. It's the different, like I said, center of mass, sum all the differences squared, and then you square root it. So it's kind of smaller than the radius of the particle itself. So don't be surprised if it's a small number. And radius of hydrodynamic radius changes depending on shape, of course. So I just thought I'd give you that just so you can see sort of what that number means. And okay, here's some more pictures of the Guinier analysis. So I'm going to stop at this point, and we'll take a little break, and then we'll continue with this after the break. Thank you. Just a little faster.
Once we get here, so as long as he's okay with that. He's actually here too. Oh, no, I mean, the funny thing is the lab, since Nozomi comes from this engineering kind of background, it was always, he'd always planned the lab to do more engineering.
I think I'm going to start again. So there's some slides I didn't show, which I'm just going to mention because we talked about radiation damage, and which is an ever-present problem in X-ray studies. And so when a photon encounters atoms, there are sort of three things that can happen. the thing that happens most often is the atom will absorb the photon, all of the energy, right? And X-ray photons have a lot of energy. Their energy is measured in thousands of electron volts. So 10 keV is a typical energy that we work at. If all of that is dumped into an atom, you can imagine what happens. It shoots high-speed electron called photoelectrons in every direction. And this is what causes damage. And this is the most, actually, unfortunately, at 10 kiloelectron volts where we work, this is the most frequent thing that happens when a photon encounters an atom. Most often, most of the time, the atom will absorb that photon and spew out a high energy, we're talking thousands of electron volts, photoelectron, which will then collide. Electrons interact very strongly with matter, as you know if you do electron microscopy, and that will collide with other atoms, which will cause more electrons to spew out, and you have this cascade of electrons floating around in solution, ripping things up. So that's damage, and that can chemically activate the surfaces of proteins and cause them to stick together. And that is the principal thing that messes up SAXS, because as you know from the formula, right, the big molecular weight stuff dominates. So as you're damaging your protein, you're starting to see oligomers form, precipitates form, and it's the end of the experiment. So that's why we flow sample through the beam instead of just letting it sit there and cook. The second thing that happens is called Compton Scatter, which we don't talk about very much. It's a small but ever-present thing, and that is an electron comes in, it's just like a billiard ball. It kicks out an electron with low energy. There's not very much of that, so you don't need to worry about it. And so those two kinds of damage, they don't produce any particularly useful information. And it's the third kind of scattering where the photon comes in, the electron acts like a ripple, a float on a pond, and it creates ripples, and no energy is transferred. That's an amazing thing. It's an amazing thing. A photon comes in, changes direction, and goes out. It's a wave phenomenon. So those are the three things. Now, as you go to higher energies, the proportion of those three things change. And so, which is why when you go to the dentist office, you don't get enough radiation damage to really worry too much about. And the reason is at very high energies, that absorption, that first step where a photon, where the atom absorbs the entire watt of energy, that becomes less and less probable. It falls really fast with energy as you get up 10, 20, 30, 50 keV, the kind of things that your diagnostic X-rays. So that's just so you know, that's a paradox. You can explain to all your friends the paradox of why high energy X-rays are less dangerous than low energy X-rays, right? You know,
so Richard,
yeah.
are you going to discuss why typically the SAXS beam lines are set up at like 8 to 10 keV and not higher or lower?
Oh, yeah. So we can actually do, that's a good, I have a little toy on the net you can play with where you can punch in any energy you want up to about 30 keV and see what the SAXS experiment would look like. So as you get to higher energies, so most beam lines in the world operate at either 10 to 12 keV. And those are energies, crystallographers are used to maybe 12 or 13, and that's made, I think originally the SAXS beam lines like the low energy because it makes it easier to get to lower angles when you have a low energy. But it does, your samples have more absorption, and so you also can get optimal signal from a shorter path length, it turns out. So those may be the reasons why we operate at 10. Svergen's line operates customarily at 10, I think, and but you may go to others that operate at 12. Nobody goes to 20. We've done that experiment. It becomes harder and harder to get low angle and easier to get wide angle. However, it is thought that there might be an advantage of lower radiation damage. At least that's what the numbers suggest, is that as you go to higher energy. But it's not customarily, of course, this is a high energy synchrotron source, so we can produce very bright high energy photons. But customarily, you're working around those wavelengths for that reason. Also, it's tough. If you go really low in energy, air starts to absorb, and the optics are hard to do. So that's kind of the reason why we are where we're at. Okay. All right. So I talked about the Guinea analysis. This is what you will see in the tutorial, something like this. This is in the raw program when you have collected your first scattering profile. You've subtracted the buffer, you have this curve, and you've performed the Guinea analysis, right? And that plots, it makes a plot of logarithm of I, your intensity, as a function of Q squared. And it looks at the small q area, and you should get a line. You should get a nice linear thing. And you will see it will calculate automatically your radius of gyration, and it will extrapolate to 0 and tell you what the intensity under the beam stop would be. Okay. And so I'll tell you how to use those two. The radius of gyration I already indicated is the size of the particle. The I0 allows you to get the molecular weight, which is a very important step, which I'll discuss in a minute. Okay, and this is the deviation from linear, which you want to see just sort of random scattered here. Should be evenly spaced on both sides of the red line.
Just a discussion of the QRG 1.3.
Oh, yeah. So the question, because Guinea's analysis relies upon being at small angle, right? It's not a perfect Gaussian. It's only a Gaussian as you get to very small angles. So the question is, when you're doing this linear fit, how small do you have to go, right? You can see it's going to deviate it's going to bend and not be linear if you go out. So how small do you have to go? And the rule of thumb that people have used forever is when Q times the radius of gyration is less than 1.3. That's where you should start your analysis. And the only reason that this exists is that that's what, if you want, I think it's a 1% error in your RG or something. That's, oh, I'm sorry. 20, yeah, they looked at a sphere way back a long time ago in the mists of the past there. They came up with this number. And it's not a hard, but this is what all of your reviewers will look like in your, you should probably put these things in your supplementary section, and they want to see at least a plot of a Guinea, in this QRG region of 1.3 and less, that it looks linear.
It's reported in row.
Oh, yeah, QRG right here. So, this is an old picture, so there's a little more to the program now. But this, you kind of adjust this endpoint. It'll try to do it automatically until you get to 1.3, magic 1.3 number. For rods, you may have to go smaller. They don't look as linear, and then discs actually look really linear and nice. So why this is an old technique. Why do we bother to do it, right? There's a couple of reasons. One is that it's a very powerful diagnostic of whether or not your sample's good enough. So if you have a Guinea that looks like this, you are ready to proceed. It doesn't guarantee that there are problems with it, but this is a requirement of going forward. If you don't see this, you need to do some additional work. If you see that this is very common, and this is an indication that you have a mixture, some aggregates or something. You have this smile, right? This is not good news. So this is why people do size exclusion chromatography and things like that. Try to figure out why you don't have, maybe the sample doesn't, maybe it starts to aggregate, you purified it in the lab and it sat around for a couple of days. This is also pretty common. What this means is you got some dirt in the sample or maybe some precipitate. If you just spin this very fast for a long time, that will go away. We've seen that a bunch of times. This is rescuable. And in fact, we have a mini ultra centrifuge in the lab, an air fuge. They're relatively cheap. It'll spin this at I don't know what, 70,000 RPMs or something. You spin it for 15 minutes or half an hour, and this would flatten out. That often happens. So that's a rescue. That's a rescue. This is a back to the drawing board. Oh, here's an example. Yeah, this was actually this was a back to the drawing board almost. I mean, it's okay. It's sometimes you can kind of get some information from that, but see, this is the difference from the curve. You've got that smile, right? And look, the radius of gyration is kind of big. It's 93 angstroms. And watch what happens after we spun it for a half hour on an ultra centrifuge. Isn't that pretty? Look at the difference. Same sample. Look at the radius of gyration. Yeah, so there was something big dominating the signal, something large enough that we spun it down. And there's something to keep in mind when you spin your samples. If you have even glucose isomerase that's only 173 kilodaltons, if you spin it in an ordinary centrifuge for long enough, you'll see a concentration gradient. So if you're measuring concentration, just be aware that spinning it can affect the concentration. Right? Monodispersed. That's what monodispersed should look like. You can see the difference if I plot the I of Q curve, right? Look at the difference. So this is, you see this thing, you should worry. It should look like a Gaussian. Yep.
So for how long do you need that your sample is homogeneous? Because sometimes the precipitation happens during time. So
Yes, that's a very good question. The question is, how long do you need for it to be monodispersed? We've had samples that that became aggregated over a course, even when they were treated properly, over a course of a day or two. And there are samples that apparently aggregate almost as soon as you purify them. And so that's why size exclusion chromatography coupled to SAXS is so powerful, is that when you encounter a situation where your protein is not very stable and you can't keep it purified long enough to get it to the beamline, you can purify it on site. Now, it is possible to freeze proteins. Many proteins are tolerant to being frozen. So you can, the thing to do is to try purify your protein on site, and as soon as it's purified, flash freeze it. Bring it to the beamline, thaw it out, and try it that way. And then if it's aggregated, if it still looks aggregated, then you can do a size exclusion test. So
I think what Vishal would tell you to do is flash freeze it, thaw it in your lab, put it in the MAL system, see if it aggregates.
Yeah, you can test to
Then
see
if
if
it
it
doesn't,
survives
do it
freezing.
again and bring it to the
Yeah,
beamline.
I mean, the
So
team.
Thomas Grant will show you a lot of really nice looking, beautiful looking data that was all frozen from, well characterized. So
We just use a nanodrop.
Depends on how
Take a
hard
little
you
bit,
pop it.
freeze it, thaw it, and just take the absorbance again and see if the absorbance changes after it's spin.
Okay, so it is possible to publish data that looks like this, and it depends on you, but you have to be honest about it. If you have, if the best you can do and you have a little uptick, you need to report this in your paper and provide it in the supplementary materials. And in this particular case, it was not. The reviewers accepted this analysis. We could easily have done that, and it would have looked great. But that's not, you know, because we know in SAXS that small amounts of aggregation can really affect your interpretation of data. If there is any evidence at all that you have aggregation, you need to present it to your reviewers so that they know. And it's not always a killer, and it wasn't in this case. It didn't. It wasn't central to the conclusions that we were trying to draw. So that's just a lesson in presenting your data ethically. The radiation damage, like I said before, you know, you have all of these photoelectrons going around in solution, chemically activating things and causing them to stick together. I need to get a better data set. This is what it looks like if you expose lysozyme too long. You'll actually see this, that on the beamline is so hot. You'll see a much cleaner example of this in your lysozyme data sets. You know, after the first few exposures, it starts to go upwards, right? That's what your radius of gyration is increasing. The stuff is starting to stick together. And so we have tools, which you will learn tomorrow, we take a series of quick snapshots and you compare them to see when damage starts, and then you combine just the undamaged data. So we have Jesse has put in some very nice tools in the raw that will automatically let you know when there is a problem with the data, with the damage. And damage is a problem in size exclusion too. And it might be a little more pathological there too, because you need to, you see sort of baselines drifting and stuff like that. Okay, so we're coming back to this formula over and over again, molecular weight. So you all probably know the molecular weight of things you're looking for most of the time, but it's very important in SAXS to measure it, because it'll tell you if you have the monodisperse, or if you have a higher oligomeric state, or if you have some aggregation. So, and there are a bunch of ways to do this that are done automatically for you at the beamline. I'm going to tell you about those techniques. So the first one is that when we have time, I'm thinking about all the things that we need to do to get ready for tonight for you guys. We will run a water and an empty cell, and that's a good idea whenever you go to a synchrotron. And that allows you to put things on an absolute unit, in absolute units, so that I of U actually has units of inverse centimeters. And then you can calculate molecular weight from I of 0 from the Guinea analysis using a simple formula, if you know the contrast and the concentration. You have to know the concentration. So we always advise that you measure the concentration as accurately as you can. Kushal has mentioned this. And don't just use a Bradford, you know. Be careful about it, because the accuracy of your molecular weight depends on the accuracy of your concentration. And the accuracy of your concentration measurement is not very good. It's like 10% at best. So we provide this, not all beamlines do an absolute scale. We provide this by default when you arrive. We have time. And then we also provide protein standards. So we run a lysozyme, and we used to run glucose isomerase when we had it. But if you know a standard protein in similar buffer conditions, you can just take a ratio. And we do this also as part of your learning. You will take a lysozyme and you will use it as a standard to calculate molecular weight. So this is another thing that requires that you know the concentration accurately. So these two methods require concentration. And then these two methods are really pretty nice techniques that do not require concentration. Again, they are calculated automatically for you when you collect SAXS data. Jesse will show you how to pop up that panel. Well, and they both involve integrals. This is an old method called Perot volume. So the integral method, instead of doing a Guinea analysis and depending upon small q, it depends on the entire curve. And that's nice. That means it's less sensitive to damage and aggregation and aggregates. However, it means you have to go far enough out in Q. And if your curve is, if you have a small protein that doesn't fall off rapidly, that these numbers can be not very useful. They do depend upon the DNA analysis indirectly. Indirectly, yes. So these in chromatography, these are the ones that we plot for you automatically on the curve. So you get a molecular weight, but you should examine to see whether or not they're likely to be converged. Okay. All right. All right. So that was, so we talked about this parameter. Let's talk about this one for a minute, the structure factor. Most of you will be doing dilute solutions, and so you can completely ignore, well, not completely ignore this. In every SAXS experiment, you are required to demonstrate that this can be ignored. And you demonstrate that this term can be ignored by doing a concentration series, at least 3 different concentrations, to show that nothing's changing. So you're dilute enough that you can assume that this is 1. Let me see if I have a picture. Yeah, well, this is a visual way to understand what's going on. That S term is diffraction of electrons that are not within the same protein, but in different proteins. And you can see that a wave coming in, hitting an electron here, and a wave coming in, hitting an electron here, the shift in phase is really tiny. It's a small, you're seeing scattering at very small angles, right? So that's the solution part. So that's why the Guinea part of your curve is influenced. As you get more concentrated and neighboring particles are getting closer together, your Guinea analysis is starting to get messed up. And that's what you need to do a concentration series to understand. Now, this can be very useful information too, but so here's what it looks like. If you were to take a lysozyme at 1 mg per mil, you look, it's that nice Gaussian shape, it's ideal, but noisy. And then as you go to higher concentrations, you get less noisy data, but look what's going on, it's dropping. And this is very characteristic of what happens when you have what's called repelling. In other words, lysozyme has a charge on it. And if you don't have screening in your solution with salt or something, and your particles get close together, they will avoid each other. And the symptom or the effect of that is that it makes the guinea egg go down. The opposite of that is if you have clustering and your particles are hanging out close together, like aggregation, you're going up. So these are all things that we wish to get rid of. We really want this nice in-between where you're going to get a proper Gaussian curve where it levels out. Okay. But you can interpret this information, right? This is something that if you can interpret that and talk about ways to modify proteins so the viscosity and the properties of the solution are better for some reasons. So if you're really trying to get very good data, and you notice that the dilute is very noisy, see how noisy that is, the dilute, but it's correct in small angle. You can combine concentrated and dilute. People have done this for years. It's acceptable. People argue over whether it's always acceptable, but it's, see at wide angle, the curves are the same. And the reason is that out here, this is conformational change. Usually concentration doesn't cause conformational changes. It can, right? There's a phenomenon of crowding that's very interesting, but that for the most part in the regime that we're looking at, it's not close to crowding yet. And so you can do this if you,
So how much is wide angle affected by, say, aggregation? Like the fine features, for example, if you have a spherical particle.
None.
None.
Yeah. The aggregation is purely, it falls off fast with Q. Yeah. So even if you have an aggregated sample, in principle, out here is still telling you about the.
So you could combine dilute good with aggregated bad at high concentration. Theoretically.
Yeah, you might be able to do that. Yeah. You have to tell them it's aggregated though.
It would be hard to push that faster reviewer. Let's put it that way, whether or not it's kosher.
Just keep sending it in and you'll eventually get a bad reviewer. Okay, so yeah, few people do this. Usually you pick a concentration that works and you go with it. But if you're really trying to push your yeah, I want to stop for a minute. Let's see. Oh, yes. Oh, yes. So what's the optimum concentration to use? All right. Notice M times C. So if you have you want to get a certain I value that's high enough. It's got to be high enough. Then M times C are here, inversely related. So if you look at what concentration should I have to get a decent signal as a function of molecular weight, right? It's a hyperbola. So low molecular weight stuff, 4 mg per mil is great. And so you want to do you have to do a concentration series, some dilutions. So you go, maybe 4, 8, 2, be kind of your minimum. Whereas something larger like glucose isomerase at 173 kilodaltons, 0.3 mg per mil is a sufficient. And so you want to do this kind of a concentration series. So you've seen different. Kushal put up an M times C approximately 100. I have M times C equals 60. Will give you that. So 60 is the number I just picked out of the hat from what we do here. So, okay, yeah, sorry I didn't extend this out. So many people have bigger, but you can see where it's going, right? It's going to go into the point something mg per mil when you have a half a megadalton or something. Everybody following? Makes sense? Okay. All right, so what I want to do, yeah, this is a good moment to stop for a moment and go to a web application that I have. Here it is. All right. I don't know if you can see this address or not up here. You can go to this, www.class.cornell.edu/web_sax/parsing post _ sorry about that, it's a long. You can get this, you can get my SAXS calculator. And this will give you a highly realistic and validated prediction of what a scattering experiment is going to look like on the beamline. And you can enter the energy, the photon flux. This is how bright your beamline is. You ask your beamline scientist. 10 to the 12 is what we can reach on G-line. It's pretty good. You might see at the state-of-the-art beam lines like Petra 3, you could scorch your samples at up to 1 to the 14th, 10 to the 14th. Laboratory sources, probably 10 to the 8 or 10 to the 9, maybe. I'm not sure.
Is that total flux or?
This is total number of photons per second hitting your sample. And people use different size beams, but so this is like, good hot day on G1 right here. So, F1 will be in the 10 to 11 somewhere. I have a number. Sample to detector distance is usually fixed by this is how far away your detector is, but if you want to play with that number, you can. And exposure time, you pick your exposure time, you can see what it's going to be like. This is the standard little detector that we have. You can try bigger detectors, they take a little longer. Depends on what your beamline has equipped with. In principle, you can do RNA and DNA here, but it's not implemented at the moment. We don't have those parameters in there. And here we're just, you have a sphere or a cylinder, and this is the height to radius. So you can make like a rod or a disc. So that's very simple. And then you punch in what your molecular weight is and your concentration. And ignore this for a minute. Everything is in 150 millimolar salt, whether it likes it or not. And you hit return. So this is what a lysozyme at 4 mg per mil, assuming it's a spherical lysozyme, looks like. See? So remember the theoretical has the bounce in it, but you have this noise. this is the Q range that you will get. Here's what your guinea A will look like. Okay, this is a crack Q. We'll talk about that later, and some other parameters. So now let's play with some things. What will happen? Somebody tell me if I go up to half a megadalton, what's this curve going to look like? Is it going to be broader or is it going to be steeper? Steeper, right. It's going to be a lot. It's going to fall off faster. Yeah, let's see what happens. And we're going to stick with 4 mgs per mil. So let's go ahead. All right. Nice data. That's a nice guinea egg. Look at that. But you know, if you look at, if you zoom in, you look at the difference there, you can see that there is a, even with a pure spheres, remember, guinea egg approximation is a small angle approximation. So now this is a tiny difference, right? This is a really good guinea egg. And look, this is, so that would be kind of what you would see with a virus particle. Yeah. And you can see that if we go down to, you know, you can probably get down to 0.1 mg per mil is yeah, it's still looking pretty good at 0.1 mg per mil. Although, see how quickly it falls off and you lose data. You'll notice that you're only getting out to 0.1. By the time you get out to 0.1, you know, you've gotten kind of noisy. You've lost a lot of, you know, that's large particles for you. But there's a lot of signal here. So, yeah, and you see the, okay, so you can play with that and see with different exposure times and stuff like that. Oh, and energies. All right. E, what energy should I look at?
A1 still 20?
A1, 20, we can go to 20 keV. Yeah, okay, let's see what it would look like at 20 keV. Okay. So, yeah, it's a bit noisier. Yeah, a lot more scattered. Now, in here, I assume that you can measure all the way down. I don't try to predict, compensate for the fact that it's harder to get low angles here. But you can see that on that same detector, you're getting big wide angle at 20 keV, but you're not getting much signal out there. So, okay, so have fun with that. We'll continue to improve that toy. All right. So the last thing I want to talk about here is contrast. So this term, this rho 1 minus rho 2, right? So rho is the technical term for it is the scattering length density. It's really electron density, average electrons per unit volume times a constant, which is the scattering length, but you don't need to know about that. So, and this is the particle itself minus the solvent, right? So the solvent, you can easily imagine, oh, I can easily calculate the number of electrons per unit volume, right? That's a undergraduate chemistry exercise. For a protein, it's a little trickier to come up with that number. Of all protein, there is a way to do it, and you can look in the paper that there are actually programs that do this. The neutron people have to estimate these kinds of things frequently, but but basically the idea is a protein does have a number of electrons per unit volume, and here it is. It varies, of course, but 420 electrons per cubic nanometer. And look at water is 330, so they're pretty close numbers, right? And looking back at that formula, see it's the difference of the square of the difference. So that means that you can see proteins as kind of weak, it weakens them. Look at nucleic acids, really strong because of the phosphoruses are pretty electron rich. Look at lipid. I need a drink. Hot water. So what happens if I'm measuring a protein in 65% sucrose solution? What's my scattering going to be? You see the trick? You see what's going on? If I have a buffer that has the same electron density as a protein, I will see what? Nothing. Nothing. Now that's a trick that you can use, contrast, and I think that Steve is talking about it. No, not Steve. Vishal is talking about it.
Yeah, one example from here at CHESS.
Yeah. So you can do this to make certain things disappear and certain other things stand out. It's a trick the neutron people do a lot and can do more easily, I think. I don't know, is it more easy?
It's a wider range of contrast with neutrons.
Yeah, more flexibility, more versatility with neutrons doing this. But it has been used successfully by several groups here, and if you have mixtures of DNA and protein and so forth, okay? But the lesson for this though is that for normal SAXS, you don't want your buffer to be anywhere close to your protein in terms of density. So try to keep your glycerol low. Having a little glycerol is a good idea, 1 or 2%. Keep your salt solution low, less than 1 molar, generally. And avoid lipids and detergents and detergent micelles unless you know what you're doing. Because look at lipids. That's a challenging problem, doing membrane proteins and halos. People do it, there's been success, but you need to know what you're doing. It's not a good first thing to try if you have lipids or detergent in your solutions. Okay. I think that is, hang on, I have a couple more. Let's see, I go until what? Yes, I have one more short bit to talk about, and then I'm done. And then we're going to do, they've asked that we do the you're right. They would like for us to do the group photo immediately before lunch. So, all right, so I mentioned at the beginning of this talk that one of the really cool things that SAXS can tell you about is disorder and unfolded stuff. It can tell you things. And so remember that sphere, if the form factor for the sphere, if you plot it on a log-log scale, here are the bounces. Look at that, it forms a line. Right, the envelope has a slope that's always exactly minus 0.4, minus 4, q to the minus 4. Okay, is how, this is called Perot's law, and it's a general result of a polymer, the theory in the field of polymers, that if you have a folded object tightly compact with a well-defined boundary between buffer and protein, that the curve will fall rapidly with wide angles, like q to the minus 4. So pretty quick fall off, not exponential though. But unfolded random walk polymer has a lower power, and a fully extended chain, like if you put some, if you're going to do a gel, and you're denaturing gel, you get a very low fall off, a linear. So there's a special plot. They're so easy to do. It's called a Kratky plot. Q squared times I. And if it's folded, you'll see a single, or you might see several humps, but it's going to fall down to 0, right? Because it's falling off as a high power Q, and you're multiplying by Q squared. So you can tell if something is folded or partially unfolded, see? By doing this Kratky plot. Very simple plot. You'll learn how to do that in the, it's just an option, plot option in your software. Can anybody guess what common problem in sample preparation could really screw up this plot. Think about, Kushal was telling you about the different things in sample preparation go wrong. Is this plot depending on small angle stuff or wide angle stuff? Okay, so it's depending upon the wide angle stuff being accurate. Okay, and if you don't, let me see if I have it on the next page. No, I don't. I should have a diagram, I should have a picture here. So in order to get this part of the curve right, you have your subtraction, your buffer subtraction has to be right. And if you don't have a buffer match, if you don't have a good buffer match, your data out here is going to be artificially, you know, it might go negative if you have a crossover, or it could look like that. It could be a buffer subtraction error. So that's why you want to make absolutely sure, if you're going to use this to interpret folding, that your buffer background subtraction is good. Okay. That's a common test question when they make up exam questions.
Yeah, question back
I have a
over
question.
Yes.
So if you didn't buffer match perfectly, would it still go towards an asymptote, let's say? So if you didn't buffer match and you were still subtracting the same wrong buffer, wouldn't it still go towards one flat line as you went out?
Well, let's see. It's like adding a constant. Like so instead, you mean it might look like that, I think, right? If you're adding a constant, so you might confuse partially folded with folded if you had a mismatch. Now there's a trick. Well, I'm not going to talk about it, but there is an analysis. Let's see. You can calculate this Perot exponent. There's a very nice program called Scatter, and there's the group out at Berkeley, the light source there, have Rob Rambo and company. He's now in Diamond, I guess. Have a way of analyzing these curves at a relatively low angle to figure out what this power is. And that can tell you, and that might be less sensitive, maybe, but it's just a warning about this technique. Okay, makes sense. It's just a simple plot. This SAXS theory is so cool. It's just all these simple plots. So, is there any way to get electron density? And remember, I've been telling people for a little while. Oh well, we're not quite ready to get to that yet. Yeah. See, actually, let's see. Wait a minute. Yeah, so you are going to learn that this. I used to have to say this, that yeah, there were a few people in history who used symmetry and so forth, but Thomas is going to tell you later today how to get real electron density. You know, this is like the Professor Guinier all over again is you know, the community was trying to do electron densities and it was just really hard, and they got the bead models seemed better and they gave up on it, you know, and now we can calculate densities for real. So a crystallographer, you know, the first thing that you might try is, if you don't have any phases, well, okay, just do a Fourier transform of your intensity anyway, right? That's a P of R function. And it's called the Patterson function in crystallography. It's of marginal importance in crystalography. Crystallographers use the Patterson if they're doing molecular, if you're looking for heavy atoms and you have a small number of things, you can figure out the structure and then use it to phase things. It plays a minor role. In SAXS, the P of R function plays a very important role, and it's extremely useful. And you can get your radius of gyration and your I of 0 taking integrals instead of doing a Guinier analysis. That's cool. Less sensitive to interparticle interactions, less sensitive to aggregates. It does a bunch of nice things, and a lot of people put a lot of effort into it over the years. And it's an automatic thing that your software will calculate. But the P of R in and of itself is quite useful to plot and look at. I'm not going to bother with that slide or that slide. So this is how to think of a P of R function. Suppose you have a molecule here, and you think of all the electron pairs, right? Every possible pair of electrons separated by all the different distances. You take those and you make a bin histogram, right? And you multiply by R squared, so it goes to 0. That's what this is. That's what the P of R. So this is distance here, and this is R squared weighted probability or frequency of occurrence. So it's sort of like an electron density. It's telling you about the electron density. It's telling you about sort of the distribution of pairs of electrons in your system. And this, you can see this is a 2-domain system, and you got a little hump, a shoulder here. This is from a very old book. This is sort of the idea that you're starting to see kind of how domains are positioned in your system. This is a practical example of a muscle protein in which, depending on whether you had ions, calcium ions present or not, it existed in, this was a compact state, and then in the presence of calcium it opens up. And look at that, it's bimodal. That's cool. So this is just a great diagnostic for when some conformational changes are happening in your system. And again, it's pretty easy to calculate. These things pop out. There's a couple different ways of doing this in raw, and you will learn how to do those tomorrow. And these curves are input to a lot of advanced techniques, like atomistic modeling in envelopes and stuff like that. This is an input. So this is kind of something that you have to do as step two in the process. All right. So we're at an end of the basic SAXS that I wanted to give you a foundation so that you know how to collect the data. You know that there are a bunch of simple things that you can do right away with SAXS data, and you know what you know to be confident or suspicious of your SAXS data. So, are there any questions before we break? Or we need a group photo. Yes.
I'm wondering if there's a generally thought like lower size limit for what proteins you look at and the information you can get like once they get too small.
That's
and
a
you're
very
getting
good
into like
question.
the peptide range.
Yeah, you notice that as proteins get small, you could try out my calculator and put smaller and smaller numbers and see what happens. So what happens is the curves get broader and broader. So if your detector is fixed in one spot, then you're starting to miss more and more of the curve. You'd want to push your detector closer to get more of the wide angle stuff. And also the signal, because it's I of Q is proportional to molecular weight, the signal is getting weaker, so you have to have a higher concentration of sample. And so realistically, once you're down in the single kilodalton size, it's starting to be harder and harder to see stuff. So SAXS is not very good at small things. It's really great at big things.
I've gone as low as 8 kilodaltons on a synchrotron.
8?
With 8.
8, okay.
A monomeric Tudor domain, and that was low. I mean, that was tractable, but with synchrotron, had a home source, I couldn't do that with a straight face. Also, like in the millimolar regime.
Other question?
Pretty small, just like a truncation. Larger than so.
Oh, the silence is okay. All right, thank you very much. Let's break for a photo op here.
Yeah, so we'll do a quick photo out here right on the balcony, and I think we're just gonna miss the rain. So just kind of, I'll try to get the lake and everything in the background. Please. And the best way to get out there is just follow me this way.